Hello everyone, today we're going to be talking about the Calvin cycle. This is the second step in photosynthesis and is what happens after the light reactions. This is where those 9 ATP and the 6 NADPH are going to get used. So let's start with the first part, also Calvin cycle happens in the stroma. And the whole purpose of it is to make glucose for the plant, or sucrose and sugars like that, for the plant to grow and make fruit and flowers and things like that. So, the first step in the Calvin cycle is the carbon fixation phase. And in the carbon fixation phase, we're going to take 3 molecules of carbon dioxide and 3 molecules of RUBP, and with the help of Rubisco as our enzyme, we're going to quickly form an intermediate compound, which is this orange thing right here. And quickly, that orange compound, it's not really important because it quickly gets split up into 6 3-phosphate glyceride molecules, or glycerate molecules. So we created 6 molecules, and the molecule is called 3-phosphoglycerate molecules. So now we have 6 of those 3 PGA, which has 1 phosphate. And then the second step is called the reduction phase. And in the reduction phase, we're going to take those 6 3-phosphoglycerate molecules, and we're going to add a second phosphate to them, right? We're going to give them each another phosphate. And in doing so, we're going to take 6 ATP and oxidize them to 6 ADP. So that's why we need those 6 ATP, because we're going to steal a phosphate from each of those ATP and give them to those 6 3-PGA molecules that we created before. And now, since they have a second phosphate, they're going to be called 1,3-biphosphoglycerate molecules, and we still have 6 of them. And then, now it's on to the NADPH. They then get reduced to 6 G3P molecules, and this is with the help of 6 NADPH, and we are going to oxidize those 6 NADPH and create 6 NADP+. And the compounds from those are going to help transform the 1,3-biphosphoglycerate molecules into the G3P. So with the help of 6 ATP and 6 NADPH, we were able to oxidize the 6 molecules of 3-PGA eventually into 6 molecules of G3P. So this is important, the G3P. This is what we want. This is the whole point of this reaction. G3P is something we can work with. So we created 6 G3P. And out of those 6 G3P, 1 G3P is actually going to branch off and leave the Calvin cycle. Now this 1 G3P is a lone soldier. It's going to be used to build sucrose, to build cell walls, to build fructose, to build glucose, to build sugars, to build flowers, to build fruit. It's basically going to get used for the plant's own benefit. So this is where you see growth, from this 1 G3P. And if we redo this cycle a bunch of times, that 1 G3P adds up, and now we have a bunch of G3P building our plant. Anyways, that 1 G3P leaves the cycle, and the remaining 5 G3P remains in the Calvin cycle and goes on to the third and final phase, which is the regeneration phase. And it's called the regeneration phase because we're going to take those 5 molecules of G3P right here, and we're going to regenerate them back into 3 RUBP molecules. So 5 G3P molecules become 3 molecules of RUBP. And we're able to do that because 3 ATP get used up, and now they're just 3 ADP. So remember, in the light reactions, why we needed a bit of extra ATP? This is why. It's for the regeneration phase. And this is so we can take those 5 molecules of G3P and regenerate them back into 3 RUBP. And actually, we're back at the carbon fixation step. If you remember at the carbon fixation step, what did we take? We took 3 molecules of CO2 and 3 molecules of RUBP, which actually we just created from the last step. And with the help of Rubisco, we went back and redid the whole Calvin cycle again. So the whole benefit of this is that 1G3P slips away every time. And since we create 3 RUBP in the regeneration phase, and we use 3 RUBP in the carbon fixation phase, we can just keep the cycle going on for a very long time. And as long as 1G3P slips away every time, we can use that 1G3P to make things for the plant. And that's where you see things like growth. And that is basically the Calvin cycle right there. Again, this happens in the stroma. This is the second half of the photosynthesis reactions, whereas the first half is the light reactions, and I actually made a video on that as well. And in this process, a total of 9 ATP were used, 6 ATP in the reduction phase and 3 ATP in the regeneration phase. So 9 ATP total, and 6 NADPH were used, and they were all used in the reduction phase. So in total, in the Calvin cycle, 6 NADPH and 9 ATP were used, and the byproducts we have left are 9 ADP and 6 NADP+. And of course, the one benefit is the 1G3P that we get to create and gets to leave the cycle, and the remaining 5G3P goes back into the cycle. Thank you.